Hi everyone, my name is Valerie and I'll be sharing the diversity of fungi and slime molds that I sampled in a forest for a year as part of my undergraduate thesis work. So I first wanted to highlight why biodiversity inventories, there we go. I wanted to highlight why biodiversity inventories are important and why I sought to complete one. Since the first fungus was added to the IUCN Red List in 2003, the number of fungal taxa recognized as threatened has been rapidly increasing. Invasion ecology is also a discipline that's very understudied in the realm of fungi, and most attention to invasion ecology has been directed towards non-native plant pathogens, such as the chestnut blight that many of you might be familiar with. What's much less understood is how non-native fungi impact native fungi in the same ecological niches. In comparison to other groups of organisms like plants and animals, macrofungi and slime molds are poorly described. Only about 3 to 8% of total estimated fungal species richness has actually been named. Adding molecular data to define species is also a relatively early and ongoing process. And in North America, there's historically been a misapplication of European and some Asian taxa that's still being delineated through collection and genetic sequencing. Additionally, the fairly ephemeral sporocarps and complex life cycles of these organisms can present challenges for surveying. And while I've seen bracket fungi sporocarps persist through several winters even, the sporocarps of many mushrooms degrade within days or even hours, like some coprenoid fungi. Biodiversity data can be stored in databases for both scientists and hobbyists alike, and one that many of you might be familiar with already is iNaturalist, and it's the app that I uploaded my observations to for both this project and, of course, for my personal observations. Mushroom Observer is another site where people collaboratively post and discuss identifications, and the Global Biodiversity Information Facility sources from iNaturalist, as well as other historical and online records. And for one specific to my survey, we also have the Maryland Biodiversity Project, which records the biodiversity of organisms in the state of Maryland through vetting iNaturalist observations that were made in the area. So when you think of a mushroom, it's the reproductive part of a fungus and just one part of its life cycle, much of which is out of sight embedded in organic matter as mycelium. Macrofungi are members of the Basidiomycota and Ascomycota phyla, which produce visible fruiting bodies, or sporocarps. Substrates are what the fungus is growing out of to obtain nutrients. And these pictures illustrate some common fungi substrates like fallen logs, thinner branches, and soil. They also show some of the vast diversity in sporocarp morphology, which also makes documenting particular features crucial for identifying certain fungi. Now moving on to what is a slime mold. They're fungus-like in appearance with often colorful fruiting bodies, but they are actually not fungi. And unlike bacteria, which are less specialized, a cell type called prokaryotic cells, slime molds have eukaryotic cells the way humans and fungi do. The taxonomic group that slime molds belong to is a big polyphyletic group called Mycetozoa, and the group being polyphyletic means that what we call slime molds evolved multiple times, not just once. And they're microscopic ones you need to see with magnification, but a lot of macroscopic ones you can also see with the naked eye. A lot of those bigger slime molds are in a group called the myxomycetes, and these weird and small organisms might seem easy to overlook at first, but myxomycetes actually comprise 50% of all amoeba-like organisms in the soil. So what I want you to take away from the life cycle diagram in particular is that this fruiting body stage, which is the most identifiable and conspicuous stage of a slime mold, is really just one sliver of a complex life cycle that we can't see. The plasmodium stage engulfs microorganisms for food before environmental cues then trigger the plasmodium to differentiate their cells to make fruiting bodies. In this case, a particular fruiting body called a sporangia. And this can take a matter of hours to a couple days. So here I just wanted to illustrate how different the plasmodium of a slime mold is from the fruiting bodies. This is a chocolate tube slime mold, which is very aptly named, and you can see that the young sporangium looks like spilled scrambled eggs, while the mature sporangia are more like chocolate sprinkles. And a lot of slime mold species have yellow and blobby-looking plasmodium, so that's why the mature fruiting bodies are used for identification. I luckily found this developing sporangia next to the mature ones on this log from New Jersey. So now that we're caught up on the study organisms, I would love to introduce my surveying project I conducted as an undergraduate researcher. My advisor had access to the Isaac Walton Lake in Poolesville, Maryland, where he helped me conduct 18 surveys about every 2 weeks for a year on the same trail. The short trail we followed, which was just a half-mile long, was in a second growth oak hickory forest that had both standing and decaying hardwoods, as well as some very rotted eastern red cedars from earlier forest succession. Each survey, a collaborator and I would pick a different side of the trail and scan for macrofungi and slime molds on a variety of substrates for about 4 to 5 hours. I recorded all my observations on iNaturalist, an open data resource that shows global patterns of ecological diversity using citizen science observations. I was able to identify many of my specimens during the surveys, but also heavily used different printed and online identification guides and got some great suggestions from Facebook identification groups as well. The wonderful thing about iNaturalist too is the collaboration from others to help identify your observation, and you can edit observations to add more data. For fungi that I couldn't identify, I collected the sporocarps and dehydrated them for DNA barcoding, which was supported by MAW DC's undergraduate research grant. So the questions that guided my survey were which species of macrofungi and slime molds are found in a single field site. I was really motivated by the lack of macrofungi included in conventional biodiversity inventories. And how do precipitation and substrate type influence the fruiting body occurrence and species richness of macrofungi and slime molds? So from my 679 observations of macrofungi throughout the year, I recorded 167 species or morphospecies across 86 genera and 57 families. This graph is called a species accumulation curve, and it shows how many different species are recorded at the field site with every subsequent survey. You can see that the number of species found at the field site is continuing to increase with every survey and hasn't leveled out yet. This means that I definitely haven't come close to recording every macrofungus on the trail, which makes sense from my own observations on hikes and other literature that find that macrofungi taxa often fruit sporadically from year to year. I actually read about a 5-year study where only 13% of the total taxa recorded in the field site were represented in any given year. So different functional trait frameworks have been applied to fungi. I categorize my macrofungi observations as lignicolous, terricolous, mycorrhizal, or parasitic. Lignicolous refers to fungi that grow on wood, whether it's saprotrophic on decaying wood or parasitic on a living tree. Terricolous refers to taxa that fruit on bits of forest floor detritus like small sticks and fallen leaves. Mycorrhizal taxa are fungi that have a symbiotic mutualism with certain plant species in which they exchange nutrients through the fungal mycelium and the plant roots. Most of the fungi in my survey were lignicolous and occurred on decaying hardwood. Many common taxa with persistent sporocarps and a longer seasonal range were in this lignicolous group. And even though most lignicolous and terricolous fungi are thought to be saprotrophic, many species are parasitic, some only facultatively so. So there's a lot of different ways to interpret and delineate the life histories of fungi that can sometimes overlap. So I created a separate category for the few parasitic fungi I recorded in the survey that don't break wood down. Mycorrhizal fungi represent such a small proportion of observations due to their narrower seasonal window compared to the other fungi. Some taxa found on wood exhibit a preference for substrate size. Fine woody debris refers to wood that is less than 10 centimeters in diameter, like twigs and smaller branches. Some taxa, such as this hexagonal polypore mushroom, were exclusively found on fine woody debris, whereas almost all of my slime mold observations were found on coarse woody debris, which are logs larger than 10 centimeters in diameter. And these substrates can hold a lot of moisture, which seems to favor slime molds. I used an online weather service called Weather Underground to record the amount of precipitation that occurred in the area a week prior to each of my surveys. I found that the amount of precipitation could explain 42% of the variation in species richness for macrofungi. But I did not find that precipitation 1 week prior to the survey was predictive of slime mold species richness. A major contributor to this is the temperature dependence of slime mold fruiting. In temperate forest ecosystems like the one I surveyed, slime molds do not produce sporocarps pretty much after November when the temperature drops. This is what I observed both in my survey and in seasonality data for the Maryland Biodiversity Project when I looked at their records of slime molds. And so in months with high precipitation like December when it snows, there were no slime molds to be found due to the low temperatures. In addition, there are some observations in the literature about the inhibition of sporocarp formation due to heavy precipitation. So the physical disruption of the raindrops can prevent the sporocarps from forming for slime molds. I did observe this in one of my surveys when there was quite a bit of rainfall a week prior to a survey in early fall. And this yielded high macrofungal species diversity, but I found very few slime molds that day. And that is sort of a contradiction to what we might expect, that slime molds are typically found on coarse woody substrates that can hold a lot of moisture, but it turns out that recent precipitation is not all that predictive of their fruiting. There was a lot of variation in precipitation, especially given the summer drought we had in 2023. I used Weather Underground again to find the amount of precipitation for each month of my survey, depicted in blue, and compared it to the average monthly precipitation for the previous 5 years, which is in orange. My first surveys in June started out very slow, and you can see that there was less precipitation than in previous years. This was more pronounced in August, which had much less precipitation than usual, and I found a sharp decrease in fungi species richness as well, as the total observations before the more speciose surveys that happened later in the fall. October had distinctly less precipitation than usual as well, and not that many less species recorded, but when I used iNaturalist to compare the number of fungi species others recorded in Maryland, there was an increase in species richness between September and October. So while more years of data are needed to see trends, it is interesting so far to consider how drought affects fungi. Here we have the 5 most frequently observed fungi families from my survey. So from the left, Styriaceae contains the several species of Styrium I found. A lot of the Polyparaceae were Trametes species, including this turkey tail, Trametes versicolor. Schizophalaceae is a very small group in general, but I had so much Schizophilum commune in my survey that it was one of my most observed families anyways. It's also known as the split gill fungus, and one of my favorites to photograph. Moving on to the Mycenaceae, most look like these clusters of tiny gilled mushrooms, and I did find a lot of these in my survey, but the most commonly observed member of this family I saw was actually Pinella stypticus, which I can show a picture of in a bit. And lastly, finding so many Urpex lacteus made the Urpicaceae a frequent family to find too. Most of these commonly found species are bracket fungi that have tough sporocarps that can last throughout the year, including winter. Other fungi species have a narrower window of making fruiting bodies, like a few months or less, and some fruiting bodies grow and collapse in hours, so this can really influence which species we're biased to easily observe. So here are some more frequently encountered fungi in my survey. As promised, I have a picture of Pinella stypticus on the top right. It was really funky for me to find out that this was in the same family as those soft Mycena mushrooms, since their sporocarps are so persistent throughout the year. You can see with the scale of my finger just how small they really can be. A neat fact about them is that they're bioluminescent, meaning that they produce light. I haven't seen it myself personally, but I've been told that others have had better luck with fresher specimens and using total darkness. On the top left, I wanted to show off Phlebia incarnata, that pink bracket fungus, which often appears with Styrium fungi that you can see in the picture as well. Some people suspect that Phlebia incarnata is parasitic on the Styrium. I haven't seen any scientific writing documenting this, but it's definitely interesting to consider with the observations that P. incarnata is almost always found near Styrium, and other mushrooms certainly do parasitize Styrium. I also wanted to highlight the species because I just think the pink color and wrinkled undersides are interesting features seen among the other bracket fungi that are typically found on rotted logs. On the bottom left, I have pictures of Gymnopus dryophyllus, which is a common mushroom that grows in forest litter like leaves and twigs. The species name Dryophyllus is Greek for oak-loving, but they can be found in mixed hardwood and conifer forests as well. The sporocarps of this fungus aren't as tough and persistent as the previous 2 species, but they fruit from spring to late fall, so they appeared in many surveys. Finally, we have Auricularia angiospermarum, otherwise known as wood ears. You might be familiar with the popular Asian species that are sometimes used in soups, but this is an American species that fruits on the wood of flowering hardwood trees, which is what the name angiospermarum refers to. There are other wood ear species which grow on conifers, so this highlights an example of how important it is to document the type of wood you see fungi attached to, or the trees around fungi that seem to grow from the ground. All of these species are typical of hardwood tree forests. So Martin beat me to this earlier, but these are two mushrooms that are commonly found even when there aren't a lot of other guild mushrooms outside from spring to late fall. On the left, we have Megacalybia rhodmani, the eastern platyful, and on the right, Pluteaster venus, which are known as deer mushrooms. And these mushrooms look pretty similar from the top, as you can see, but I usually find that M. rhodmani has streaks on the cap that radiate from the center, and I find that the deer mushrooms are less streaked and have almost like a sheen or like a shininess on top of the cap. But the cap appearance for the Pluteus mushrooms can be quite variable, so it's a lot more reliable once you flip them over, as the last species has white gills attached to the stipe with white-colored spores, whereas a species on the right, the deer mushrooms, they have free gills, which you can see the gills end before they touch the stipe, and they also are pink in color from the pink spores. The gills can look white in some very young specimens, but that naked ring around the stipe where the gills don't attach is a good indicator. Next, I wanted to share fungi that I've collected from leaf litter, which are the dead leaves that fall on the ground and fine woody debris, which, as I mentioned before, is dead woody material less than 10 centimeters in diameter, like twigs and small branches. A lot of the fungi that exploit these substrates are very small, with stipes just millimeters thick, and I wanted to showcase all the wonderful things that you could miss if you aren't looking for them. On the top row, we have 3 mushrooms that grow on hardwood leaf litter. The first 2 species also grow on small twigs, but the species on the far right, Marasmius capillaris, only grows on leaves, especially oak leaves. Another feature to note is how mushrooms like Marasmius solivanti in the middle have fluffy white basal mycelium, while the species in the pictures on either side of it have stipes that seem to directly insert into their substrates. I tend to find Tetrapyrgos nigripes in large patches of leaf litter, several square feet in size, with dozens of fruiting bodies, so they can actually be quite easy to spot from a distance. On the bottom row, we also have Mycena hymatopus and Marasmius rotula, which are both on hardwood. Mycena can be hard to identify, but this one is distinct in the way that it has a reddish color and exudes a dark red liquid when you break the fruiting bodies. A different species on conifer has similar characteristics, so for this one it was important to note whether it was on hardwood for identification. And a less common substrate that was specific to my field site were hickory nuts, shown in the last picture on the bottom row. Again, with how tricky mycenas can be, I was really lucky to find this attached to its substrate to confirm it is a species that only grows on hickory nuts, the Mycena crocea. There were so many fallen hickory nuts in this forest that I'd often see them covered in white mycelium, and I was really excited about finally being able to record the species when I found this fruiting body. So I wanted to show off a few of the ascomycetes I recorded as well. What distinguishes them from basidiomycetes is that instead of having basidiospores born from basidia, they have ascospores that are contained in a structure called asci instead. And the shape of the asci is why they're known as sac fungi. So even though there's a lot more ascomycete species than basidiomycete species in general, very few ascomycetes comparatively were recorded in the survey. Many ascomycetes are lichen symbionts, which weren't surveyed, and many ascomycetes are also microscopic. Most of the macroscopic ones are still quite small, the largest ones being things like morels and truffles. So while they're undercounted in my survey, there's still some cool specimens to share here. All of the fungi in the top row have a fruiting body called an apothecia, which is a cup-shaped fruiting body. The Helvella on the bottom row also has an apothecia, but you can see the shape is a bit different, like a saddle there. The last 2 species on the bottom row don't have apothecia, but instead have perithecia, which are flask-shaped structures containing a pore for the ascospores to be released. For these species, the perithecia are embedded in the fruiting bodies, so that the bumps that you can see on the surface of Hypoxylon fragiform there are where the perithecia opening forms and ascospores escape. The fruiting bodies are actually usually pink when fresh, but this is an older specimen. You all might have seen Xylaria polymorpha before, or aptly called dead man's fingers. The scientific name has been applied to a lot of fungi that look like those, but have different microscopic features. Most of the ascomycetes in my survey were actually some kind of Xylaria. but really hard to differentiate with macromorphology. I definitely encourage others to record ascomycetes as they're under-recorded and studied. I also observed several plant pathogen fungi, which are fungi that infect plants to feed off of them and cause disease. Typically, these species are pretty host-specific, meaning a particular species only infects a certain species or a few species of hosts. Apiosporina morbosa is called the blacknut fungus, which infects both wild and cultivated cherry trees. And all of the ones I observed were found on black cherry, a native cherry species common in the forest understory at the field site. The fungus overwinters in the knots and produces perithecia, a type of ascomycete fruiting body on the outside of the knots in the spring and summer, which releases spores. Alotus podophyllii is the May apple rust, a type of highly host-specific plant pathogen only found on May apples, which are a native plant. By mid to late spring, once the May apple colonies are well established, it's easy to find those orange fruiting bodies in large patches of the plant. And finally, we have the Gymnosporangium species. This rust fungus is heteroecious, meaning that it requires two different host species to complete its life cycle. The types of fruiting bodies formed and their appearance differs between the primary and secondary hosts in the fungi's life cycle. The primary host of this fungus is probably some kind of juniper tree, but here we see the fungus on its secondary host, which is a type of serviceberry. So Amanita is a large genus of mushrooms with an estimated 1,000 species. Thankfully, there's a lot less of them than that you can find around in the area. And they can mostly be found from July to October, and most of the species are mycorrhizal, meaning they rely on certain trees to exchange nutrients with. Something distinctive about these mushrooms is that they have a universal veil that covers the entire fruiting body before the stipe and gills break open and become visible. The leftover parts of the universal veil are what make the warts appear on the caps of some Amanita as well, and the vulva some have at the base of their stipe. Some also have a partial veil that leaves a ring near the center of the stipe. The gills on the underside tend to be white or pale yellow with a white spore print, and the gills are described as free, but they also are often narrowly attached, so they don't always have as distinct of a bald ring around the stipe the way Pluteus mushrooms do. You can see the free gills well, though, in the particular specimen I have of Amanita russuloides on the bottom row there, which you can see the zoomed in underside. So I wanted to go over examining some of these features in my collection. As you can see, we have quite a diversity in size and morphological features here. Lots of Amanita are shades of white and brown, but also some are yellow and orange and red. And I found most of my specimens in September. When identifying Amanita, it's really important to dig down carefully to examine the base for a bulb without damaging it. You can see in the photos of that Amanita section phalloidii and Amanita rhopalopus that the bulb at the base of the stipe is well below where the mushroom is popping up on the ground, so you definitely have to do some careful digging around it. There's a lot of different types of bulb morphologies presented here. In something like Amanita crenulata, you see that the bulb is quite round and globular compared to something like the specimen in Amanita section validi. And the Amanita russuloides, which don't really have nearly as enlarged of a bulb, and they also have a thin ring of universal veil fragments, where the top of the bulb meets the stipe. On this Amanita mushroom in the section phalloidii, you can see that the bulb at the base looks like a cup that the mushroom is sitting in, and this feature at the bulb is specifically called a volva, spelled V-O-L-V-A. And you can also see the mushroom has quite a large annulus or ring hanging around the stipe that just freshly broke away from the gill surface. And that's a distinctive feature as well. Not all Amanita have a ring, or the ring might often be degraded or fall off. Amanita rhopalopus at the bottom has what is described as a dog-legged bulb, which is kind of a funny descriptor, but it describes the way that it often curves to one side as an elongated oval-shaped bulb. Amanita coqueri has a distinctively large bulb with several distinctive rings of recurved scales, meaning that the scales are curving outwards. And you can also see that a lot of these mushrooms have warts, some white, and there's one with also yellow warts. The last interesting thing is that smell is often another diagnostic quality to note for a lot of the Amanita mushrooms. A lot of mushrooms in general admittedly smell pretty similar to me, but some like the Amanita rhopalopus really do have a strong smell, in this case a chlorine smell. It turned out to be kind of characteristic of the species, which Michael Kuo from Mushroom Expert describes as chlorine or rotten meat smelling. My collaborator thought it smelled like stinky cheese, so it's also cool how observers can differ in experiencing the smell sometimes. Next, I wanted to go over some boletes from my survey. Boletes are a name for a big group of mushrooms in the order Boletales, most of which are mycorrhizal. All of these species associate with hardwood, especially oaks, that I collected, with the exception of Pulvera boletus, which is more broad and associates with conifers too. Most boletes have pores on the underside of the cap, and the size of the openings and the depth of the pores are often diagnostic features. So cross-section pictures, like the ones that you see for Tylopilus rubruberneus, are helpful for measuring pore depth. Bolete spores come in shades of brown, olive, yellow, and pink, and the caps are quite substantial, but you can see there's a large variation in size, especially with the shape and the width of the stipes, if you just compare something like the Tylopilus to the Lecanellum. For Pulvera boletus curtisi in the top row, some distinctive features are the slimy cap, and the large amount of white basal mycelium at the bottom of the stipe. For some specimens, tasting through a chew and spit test can also be useful for identification. I knew this Tylopilus was going to be bitter, but I still wasn't prepared for how long it lingered in my mouth after. It was truly one of the worst things I've ever tasted. But now I will never forget that one. And there's also a lot of species that stain blue, like the Cyanoboletus cyanotinctus. This species is quite dramatic in the color change, but others are more subtle. When you injure the flesh of these blue bruising mushrooms, especially the pore surface and interior, the blue colors formed from the oxidation of chemicals in the fruiting body when they're exposed to air. Boletes can feel daunting to identify, but there's resources like the bolete filter online, which can help you sort through options by region and macromorphology. For the family Russulaceae, it mostly contains the russulas, which are called brittlegills, and the milkcaps, which belong to a couple genera. Milkcaps get their name from the white-colored latex that exudes from their gills when you injure them, which you can see in my picture of the lactiflu species. Some of the milkcaps have a fishy odor as well that's super distinctive. Brittlegills and milkcaps occur from May through October, and I found that my observations were especially prolific in the late summer. Most of these mushrooms are mycorrhizal, and they have no universal or partial veils, so you won't see rings or warts like you do see in amanitas. Their spores range from shades of white, creamy yellow, and orange. Russulas come in an incredibly wide range of colors, but that often doesn't make them easier to identify, unfortunately. So many Russula species in Eastern North America look very similar. So Russula parvovirescens and Russula belui are actually two major exceptions to Russulas being notoriously annoying to identify, as the green cracking pattern on the parvovirescens is pretty distinctive, and so is the scaly pattern on the cap of Russula belui. Some Russula species taste acrid or peppery if you chew and spit them, as I'm told, but I personally haven't experienced one like that yet. Identifying these with macromorphology can be super frustrating, as a lot of species described have variable and overlapping characters, like cap color and spore color. There's undercounted Russula diversity in my survey, as I found so many red-colored ones, but can't distinguish them from each other, so I've had to count them as just one species. But I've been able to do some DNA barcoding with MAW-DC to help with identification. DNA barcoding is a method of identifying species using a standardized portion of DNA. DNA contains 4 chemical bases, and the order or sequence of these 4 bases should ideally differ a lot between different species in the standardized part. So after you obtain this DNA sequence from an unknown fungus, you compare it to a reference database. Many fungi species sequence may not have a match in the database, but it can still help you compare how similar it is to other sequences from other recorded species. A few different gene regions are studied by mycologists, but a common region that is sequenced is the ITS region on the fungal ribosomal DNA. The process first starts with DNA extraction, in which you take a small piece of the dehydrated mushroom and combine it with some chemicals to break the cells down to access the DNA. This is actually a picture from one of Mount Rainier's monthly DNA days, which are open to the public, and I'd encourage you to join. So you can see that the tissue paper voucher containing my specimen that I labeled with my iNaturalist ID number to keep track of my samples. And you can also see just how tiny of a sample you need for the whole process in the bottom of the tubes, although it's always good to have more available to run failed batches or have some for other types of analyses. Once the DNA is extracted, a process called polymerase chain reaction, or PCR, creates many copies of the chosen gene region, in this case, the ITS region. Through another process called gel electrophoresis, the DNA product from the previous step can be confirmed. And then the samples are sent to a company which purifies and determines a DNA sequence before sending the data back. So this is one of several russulas I collected during my survey, which, as I mentioned, are hard to identify and not well described. When the data came back for some of my samples, Megan on the MAW-DC DNA team was so helpful in guiding me through how to clean up and compare the sequence to a database of sequences using the NCBI BLAST tool. The database provided a sequence that was 99.83% similar to a sequence described as Russula cremorosea. This isn't a well-described species, and there isn't a type sequence, but it closely matches several sequences described as a species. Other tools like microscopy and sequencing other gene regions provide more data to create robust species descriptions, but this method definitely has helped me more accurately portray species richness in the russulas. So moving on to some of the slime mold data from my survey, from 77 observations, I recorded 16 slime mold species or morphospecies across 12 genera and 8 families. My species accumulation curve below seems like it leveled out in the middle, but that was during the colder months when slime molds weren't fruiting. I recorded a species in the last month of my survey during May that I hadn't the previous year, so there's definitely some more slime molds to observe if more surveying was done. These are the most commonly encountered slime mold families in my survey. The families Archaeariaceae, Reticulariaceae, and Ceratiomyxaceae. I'm going to go through them and show the species I recorded. So these are the species of Archaea that I found, which are in a range of lovely colors. Archaea is a large genus, and a lot of these are thought to be species complexes, meaning there's a lot of undercounted diversity within these taxa. These slime molds have a fruiting body shape called a sporangium. The sporotheca, which contains the spores, sits on top of a stalk to elevate it from the substrate. The outer coating of the sporotheca is called the peridium, which you can see intact in the center picture of Archaea cinerea. The sporotheca contains capillicium, which look like woolly fibers, along with spores. Microscopes are crucial for viewing some of these features up close, but you can still see some differences in the capillicium with the naked eye. The capillicium of A. cinerea does not expand much compared to the other two species. You can see more of the woolly texture on the Archaea denudata capillicium, which remain attached to a small cup-shaped structure at the top of the stalk. Out of the 3 species, the capillicium of Archaea obvulata expand out the most and droop down from their stalk, as shown in the smaller photo. Archaea obvulata has actually undergone a taxon change and moved to a resurrected genus called Heterotrichia. A paper that came out just in the last year used morphological and genetic data to determine that Archaea is polyphyletic, and most of the yellow-spored Archaea were moved to Heterotrichia. The family Reticulariaceae is quite speciose, but the 2 from my survey were very common ones. They were Lycogala epidendrum, or the wolf's milk slime mold, and Tubifera ferruginosa, the raspberry slime mold. These are also considered species complexes with understudied diversity. Both of these form bright pink fruiting bodies, which burst with a liquid the same color as the exterior if you press on them. Over time, the insides become solid and powdery as a spore mass turns dark brown or gray. These 2 slime molds have a very different fruiting body types than the previous Archaea species, which had sporangia. Lycogala epidendrum has ithalia, which are cushion-shaped or irregularly shaped fruiting bodies, while Tubifera ferruginosa has pseudoithalia, where you can see that the fruiting bodies are blob-shaped, but their individual sporangia clustered together. The hypothallus, or attachment to the substrate, is also pretty prominent in the species, as the whitish material at the base of the fruiting bodies. Most of the macroscopic slime molds are in a group called Myxogastria, so Ceratiomyxa is kind of neat as a member of a separate lineage to that. And it doesn't have enclosed spores like the Myxogastria among the capillicium, but instead it produces spores externally on the fruiting bodies. Ceratiomyxaceae only contains the genus Ceratiomyxa, which only has a few species in it, but the two here are incredibly prolific. Ceratiomyxa porioides used to be considered a variety of Ceratiomyxa fruticulosa, but it is now considered a separate species. The fruticulosa has a lot of morphological variability, as you can observe in the two pictures. Sometimes the fruiting bodies look like single pillars, while other times the pillars look clustered and fused at the base. The middle picture is notably in a more mature stage, though. Ceratiomyxa porioides has a different looking morphology that has been described as net-like walls. It personally reminds me of a coral. I also wanted to highlight some less recorded taxa from the survey to re-emphasize the value of biodiversity inventories. After searching biodiversity databases, I found that these three taxa had yet to be recorded in the state of Maryland or only had one other record in the area. But none of these are known to be like particularly rare taxa. What might contribute to the lack of documentation for Coprinopsis lilacina is its tiny size and ephemeral sporocarps. Another tricky thing about it is that it has both sexual and asexual reproductive stages, and what you see in the picture is the fruiting body of the sexual stage. The asexual stage looks quite different with no gills, and it's much less recorded. Tricholomopsis flammula prefers to grow on conifers, which are less common as a forest type compared to hardwood oak hickory forests here in Maryland. I'm not quite sure why Lindbladia tubulina is not well recorded, but slime molds can develop through their conspicuous life stages quickly, like within hours, so that could be a contributor. Local biodiversity is definitely undersurveyed for both macrofungi and slime molds, so it's really exciting to be able to help record it. I also recorded Trametes lactinea in my survey, a non-native species known to tropical Asia. It's been spreading through the eastern United States in the last 20 years and is becoming quite common. This is a map of records from GBIF, a biodiversity database. Observations like mine contribute to important data sets for other work in studying fungal populations and their spread. Now I would like to briefly show off some cool fungi-related finds from the survey that weren't macrofungi. Sometimes my collaborators and I would see these millipedes in groups on a log, and at first you're like, oh, how cute, they're all hanging out. But unfortunately, they're already dead after being parasitized by a fungus. Fungi that infect insects are called entomopathogens, and some of them cause behavioral changes in their hosts, including this one. Typically, these millipedes are nocturnal and wander during the night, but spend their days hidden under the forest floor, hiding under rocks and logs. And when they get infected by this fungus, which is called Arthrophaga myriapodina, they are compelled to climb to a visible place higher off the ground than they usually dwell, so the fungus can disperse its spores efficiently. The specimens I saw were brittle and fell apart easily by the time I found them, but I found someone else's iNaturalist observation I have up in the middle there that shows the white fungus between the millipede segments where the spores are released. What I have here isn't a fungus, but it's a plant that derives its nutrients by parasitizing a fungus instead of getting nutrients from photosynthesis. Monotropa uniflora, also called ghost pipes, don't photosynthesize and don't have any green parts the way other plants do from their chlorophyll that's needed for photosynthetic reactions. This plant receives its nutrients from fungi in the Russulaceae family, which, as I mentioned before, are mycorrhizal fungi that have a mutualistic living arrangement with certain trees. So there's this intricate network between these three players of nutrient allocation where the fungi and the trees are exchanging resources while the ghost pipes are taking resources from the fungi. And I think this example illustrates what I find so interesting about fungi as mediators of these complex ecological interactions in our forests. So I wanted to showcase the mycological diversity of our local forest today, but I also wanted to reemphasize how our love of fungi as hobbyists and researchers can connect us to other ecological fascinations. Our shared love of fungi draws us together at MAW DC and also to the other wildlife in our forests. I have to thank my advisors, the Lil Lab, and my other GW colleagues for making the survey possible. From conducting the fieldwork to learning how to analyze data in our studio, I had many helpful collaborators. And I especially like to thank Saranella and Megan from MAW DC, who were so helpful in guiding my ongoing DNA barcoding work. And thank you to everyone for listening to my presentation. I'll be happy to take any questions now.
Thank you so much, Valerie. That was a great presentation, and we enjoyed going virtually to the Isaac Walton site with you.
Great, thanks for having me. I probably can't see the chat due to the specific screen share that I'm on. If anyone has questions out there, someone can read them, yeah.
Ashley says that was fantastic. Deborah, what do you plan to do next?
Yeah, for the survey, I'm continuing to refine a lot of my identifications, and I did make DNA barcoding seem like a tight and neat little process in this presentation, but a lot of runs tend to not go to plan, and you don't get enough DNA product. So it's a lot of rerunning samples, and hopefully being able to publish this with my advisor soon.
Wonderful. Let's see. Nice job from Mitch and Tom. So thank you very much, Valerie.